Hi friends, welcome to my YouTube channel and thanks for watching my channel continuously. This is the part 6 and last part of memory based ASRB NET 2023 question paper with explanation. In this part, I am going to discuss another 25 questions with some of the explanation related to the correct answer. Let us start with the first question, that is multi-line, sir. There are 4 options. Some genetic basis of resistance possesses horizontal resistance gene, then possesses varietal resistance gene against different strains or races of pathogen, and last option is different agronomic characteristics. Before answering this question, we should understand what do you mean by the multi-line. As we know, the delivered seed mixtures of isolines, closely related lines or unrelated lines are referred to as multilines. And a variety which is developed for commercial cultivation from any of these mixtures is known as multiline variety. The terms multilines and the blends are used as synonyms. And the use of multiline cultivar is an effective way to minimize the yield losses due to the attack of multiracial diseases. In a multiline cultivars, each component genotype has a resistant gene for a different race of a disease. All races of disease will never appear at a time, and all the genotype of a heterogeneous mixture are never attacked at a time. Multilines causes delay in the spread of pathogen in the field because the resistant plant act as barrier in the spread of a disease. In this way, multiline cultivars provide better protection from the attack of new races of the disease. And the correct answer for this question is option C, that is possesses varietal resistant genes against different strains or the races of the pathogens. And try to remember, multiline causes delay in the spread of the pathogen in the field because the resistant plant act as barrier in the spread of a disease. Next question is on microorganisms colonizing immediate vicinity of seed is there are 4 options: rhizosphere, spermospheres, fructospheres and phyllospheres. As we know, in microbiology, the phyllospheres is the total above ground surface of plant when viewed as a habitat for microorganisms. And the phyllospheres can be further divided into the caulospheres, that is stems, phyllospheres related to leaves, arthospheres related to flowers, and carpospheres related to fruits. The below ground microbial habitats, that is the thin volume of soil surrounding root or subterranean stem surfaces, are referred to as rhizosphere and limnosphere. The correct answer of this question is spermosphere. This is another question, that is PGTS method is there are 4 options: RNA interference, mass, QTL based selection and gene pyramiding. Before answering the question, let us understand what is PTGS and what is the meaning of these options. PTG means post transcriptional gene silencing in plant, which is an RNA degradation mechanism that shows similarities to RNA interference or RNAi in animal. However, PTGS is also an acronym for particularly vulnerable tribal group, which are the tribal community that are often identified by some specific signs such as primitive traits, then distinctive cultures, geographical locations, shyness to contact with the community at large and backwardness. And RNAi inference is a biological process in which RNA molecules are involved in sequence-specific suppressions of gene expressions by double-stranded RNA through translational or transcriptional repression. Historically, RNAi was known by other names including co-suppressions, post-transcriptional gene silencing or PTGS, and quelling. On the other hand, QTL means quantitative trait loci mapping is a statistical method that links two types of information. One is phenotypic data, that is trait measurement, and another is genotypic data, which is usually related to molecular marker. It is an attempt to explain the genetic basis of variation in complex trait. QTL analysis allows researchers in fields as diverse as agriculture, evolutions, and medicine to link certain complex phenotypes to specific regions of chromosome. And the goal of this process is to identify the action, interaction, numbers, and precise location of these regions. And the another option, that is gene pyramiding, which is the simultaneous selection for or interaction of multiple genes during plant breeding. And the main objectives of gene pyramiding includes, say, number one, enhancing trait performance by combining two or more complementary genes. Secondly, remedying deficit by integration genes from other sources, then increasing the durability, etc. Say, if we can take one example, say pyramiding has been successfully developed or say demonstrated in one crop, that is Oryza sativa for rice blast disease caused by Magnaporte grigia. And the reason behind of, say, utilizing this pyramiding in rice against rice blast is that to produce durable multiple resistance or multi-race resistance simultaneously. And pyramiding and marker assisted selection of the mass can be combined as marker assisted pyramiding. And another term, that is gene stacking, can be achieved a few different ways, and pyramiding is one of these methods. And the correct answer for this question, that is PTGS method, is RNA inference. Next question is on slope of disease progress curve, or DPC, is used to calculate. Out of all these four options, the correct answer is epidemic rate. For more details on AUPDC or TPC, or say endemic rate and disease progress, please see the question number 102 and 103 of my last video, that is part 5 of memory based question paper of NET 2023. This is another question. Who gave the final blow to theory of spontaneous generation in phytopathology? Out of all these four options, that is Robert Koch, Anton deBary, Louis Pasteur, and Pierre Anthony Micheli, the correct answer is Anton deBary. As we know that Pier Antonio Micheli was the Italian botanist and mycologist who born in Florence, Italy. He is known for his discovery on the existence of fungal spores, and thus beginning the process of dispelling some of the many myths about where fungi came from. And Michael is specialized in microfungi and is notable for having defined several important genera including Aspergillus and Botrytis. His greatest claim to fame was that he discovered the existence of fungal spores. And the germ theory of disease was established by Louis Pasteur in 1850 and later extended by Robert Koch in 1980, disproving miasma theory or theory of spontaneous generation. Later in 1950s, Anton deBary, while working on etiology of the blade blight of potato, has joined the list of establishing fungi are the cause, not the result of diseases. Next question is on marker assisted selection is, out of all these four options, the correct answer is reducing the time of varietal development. As you know that marker assisted selection of the mass refers to indirect selection for a desired plant phenotype based on banding pattern of linked molecular DNA marker. There are two prerequisites for mass. These are number one, a tight linkage between molecular marker and genes of interest, and second one is high heritability of the genes of interest. We have to remember that mass is applicable for genetic improvement of the plant as well as animal. Especially in plant, it is equally applicable in both self-pollinated as well as cross-pollinated species. The most commonly used molecular marker include amplified fragment length polymorphisms, or say AFLP, then restriction fragment length polymorphism or RFLP, and last one is random amplified polymorphic DNA, that is RAPD, and more than that SSR, that is simple sequence repeats or say microsatellites, and last one is SNP, that is single nucleotide polymorphism, and many more. This is another question. Role of salts in nucleic acid extraction of the viruses. There are four options. Out of all these four options, the correct answer is protein uncoating. That means salt in nucleic acid extraction of virus help in uncoating of the protein, and it makes the DNA molecule less hydrophilic, that is less soluble in water. And salt also help in detaching protein molecule from the strands of DNA and makes the extraction process more easier. Therefore, the purpose of the salt in DNA extraction is number one, to stabilize DNA after it has been separated from the other material or say protein in the nucleus. And secondly, another purpose is there that salt help the DNA to precipitate, that is to form a solid mass. Next question is on cell wall of Oomycota majorly consists of, there are four options: chitin, then cellulose, then peptidoglycan, and then mycolamine are in. And the correct answer is cellulose. As we know, the cell wall of the oomycetes is not composed of chitin as in the fungi, but is made up of a mix of cellulosic compound and glycan. In most of the cases, the fungal cell wall is made up of chitin, glucan, and glycoprotein. And chitin is an important structural element of fungal cell wall, which is present in the form of an acetyl glucosamine polymer of chitin. It provides rigidity and structural support to the fungal cell, and the chitinous cell wall also protect the cell from the desiccation. So the important thing of this question is that we have to remember that Oomycota, the cell wall of Oomycota majorly consists of only cellulose, but not other like chitin, peptidoglycan as well as microlaminarin. Next question is on autoclave helps in sterilization work on the principle of there are 4 options. Out of these 4 options, the correct answer is uses high pressure for increasing boiling point of water. As we know, autoclaving is a process that uses moist heat to sterilize equipment and supplies. The autoclave is a sealed device that kills microorganisms using saturated steam under pressure. The use of moist heat facilitates the killing of all microorganisms, including heat-resistant endospores, which is achieved by heating the materials inside the device at temperatures above the boiling point of water. In case of autoclave, we generally sterilize the material at 121 degrees centigrade for 15 minutes at 15 pound pressure. This is another question. Principle behind increasing temperature in soil solarization is there are 4 options: penetration of solar radiation, greenhouse effect, evaporation of water, trapping of heat using polythene sheet. And the correct answer for this question is trapping of heat using polythene sheet. As we know, in case of solarization, which is a hydrothermal process of disinfecting the soil paste and accomplished by solar proof, which is referred to as the solar heating of the soil in early publications. And soil solarization is relatively a new soil disinfestation method which was first described in extensive scientific details by Anton during 1976. The mode of action for soil solarization is complex and it involves the use of heat as a lethal agent for the soil paste from the use of transparent polythene traps. Next question is on foliage seedling disease in rice is there are 4 options. Out of these 4 options, the correct answer is Bakanae disease. As we know, in case of Bakanae disease, which is caused by Gibberella fusicori, the infected plants are characterized by abnormal elongation of the plant, lean and lanky growth and development of adventitious roots from the internodes. Such plants turn yellowish green in color and rarely bear panicles. And if panicle formation takes place in some plant, then it will bear the chaffy green. On the other hand, the other option like false smut, which is also known as rice false smut, which is caused by Astilezinoidea virens, which is one of the most destructive fungal disease in the rice grain countries. Astilezinoidins, which is a toxin, it is actually very much toxic to the plant, animal as well as human. And these are one of the major type of mycotoxin produced by Astilezinoidea virens. Another option, that is Woodbutter disease, which is considered as a minor disease of rice crop caused by Aphelis oryzae. Locally, it is known as Woodbutter disease or Udbatti roga, and it has number of vernacular names like Agarbatti disease, Mathapukaddi roga, Karikaddi roga, Chipsuhod rogsake, and this disease is endemic and minor importance in certain area in India. Very recently, we have reported this disease from Meghalaya. Then another option, that is blast, which is caused by Magnipror the grisea. It is also known as rice blast fungus. Rice rotten neck, rice seedling blight, leaf blast, an oval leaf spot of gremia, pitting disease, ryegrass blast, Johnson spot, neck blast, twig blast, then emochi. These are also caused by this organism, that is Magnaporde grisea, and it is considered as one of the major pathogen causing the most devastating disease of rice, that is called blast. This is an interesting question. Sinusitic mycelium is present in, there are 4 options: Mysticomycotina, Ascomycotina, Bacillomycotina and Deuteromycotina. Before answering this question, let us understand that the mycelium of Bacillomycotina, Ascomycotina and Deuteromycotina can be either accepted or the sinusitic or septate. The mycelium of Bacillomycotina and Ascomycotina are always septate. The mycelium of Deuteromycotina can be either accepted or septate. And out of all these, Mysticomycotina produces flagellated cells during their lifetime and may bear rhizoids, and they are mostly filamentous and having sinusitic mycelium. So the correct answer is Mysticomycotina. That means the sinusitic mycelium is present in Mysticomycotina. Next question is on taxon protozoa has been segregated from which group? There are 4 options: Chromista, fungi, plantae and Protista, and the correct answer is Protista. In Whitaker's R.H. Whitaker's classification, that is 5 kingdom classification, the protozoans are placed in kingdom Protista, and which later in Carl Wu's 6 kingdom or the 3 domain classification segregated into Chromista and the fungi from the kingdom Fungi and Protozoa, and Protista from kingdom Protista. So for more details, please see the book An Introduction to Fungi written by S.C. Dubé. For this question, there is Ganoderma is associated with, there are 4 options, and out of these 4 options, 2 options are correct. And now it depends on the evaluator on selecting the correct answers, either say 1 or 2 or say both. Before answering the question, let us have some idea on the options. The basal stem rot of coconut and areca nut is caused by different species of Ganoderma, which is a genus of wood decaying fungi. The disease is also known as Ganoderma wilt, Tanjavur wilt or Anabiroga. Then foot rot or the root rot of coconut as well as areca nut is caused by, similar to basal stem rot, different species of Ganoderma, which are also known as wood decaying fungi. Ganoderma or Anabi disease is caused by, that is, bracket forming fungus. The disease is also known as foot rot or LA or Inuit plague. On the other hand, bud rot of coconut and areca nut is caused by an oomycetes fungus called Phytophthora palmivora, and this is one of the most serious disease of this crop, especially in South India. Regarding the last option, that is stem bleeding of coconut and areca nut is caused by different fungi. For coconut, the fungus is Thalassaviopsis paradoxa, and for areca nut, the fungus is Ganoderma species. As already I have mentioned, the options for choosing the correct answer for this question is a little bit in confusion. According to the information I have provided, so the correct answer is A and C. So it depends on the evaluator which answer he is going to select, either A or C or say both. This is another question. Bivalves of Ralstonia solanacearum are differentiated based on Out of the four options, the correct answer is utilization of sugars and sugar alcohol, that is option B. Before going to the next question, let us try to understand the meaning of some terminology like say biovar, morphovars, pathogars and many more. A biovar is a variant prokaryotic strain that differs physiologically or biochemically from other strains in a particular species. Another terminology, that is morphovars or morphotypes, are those strains that differ morphologically. Next, a pathovar is a bacterial strain or set of strains with the same or similar characteristics that is differentiated at intraspecific level from other strain of the same species or subspecies on the basis of distinctive pathogenicity to one or more plant host. That pathovars are named as a deBary or quaternary addition to the species binomial name. For example, the bacterium that causes citrus canker, that is Xanthomonas axanopotis, has several pathovars with different host ranges. Say Xanthomonas axanopotis pathovar citri is one of them, and the abbreviation of pathovar is PV. And the type strain of pathovars are pathotype, which are distinguished from the types, that is holotype or the neotype of the species to which the pathovars belongs. So before answering such type of question, please recall the information related to biovar, morphovar, pathovar and many more. This is another question, that is image analysis of disease severity over time is done by, out of these four options, the correct answer is multispectral scanning. As we know, multispectral scanning is a scanning system that uses to collect data over a variety of different wavelength ranges, which is called MSS, that is multispectral scanner, and it is the most commonly used scanning system. There are two main modes or the methods of scanning employed to acquire multispectral image data. One is across track scanning and another is along track scanning. And regarding remote sensing, I want to say it is a technique that uses sensor to measure the electromagnetic energy reflected or the emitted by plant, soil, water and other objects. And remote sensing can help farmers and researchers to monitor and manage various aspects of agriculture such as crop type, crop growth, water use efficiency, then stress, yield and many more. On the other hand, GPS or the global positioning system, which has been utilized very effectively in precision farming, then improving farm planning, field mapping, soil sampling, tractor guidance and crop scouting, variable rate applications, yield mapping and many more. And geospatial monitoring, which is also another option of this question. It is an approaches to apply analysis to monitor environmental changes over the time, considering different fields of research, including computer science, remote sensing, ecology, environmental science, life science, geography and many more. Rate of inoculum increase for lead blight of potato. There are four options. Out of these four options, the correct answer is 0.3 to 0.5 unit per day. So we have to remember that rate of inoculum increases for polycyclic disease, that is the disease like lead blight, stem rust, it is 0.1 to 0.5 unit per day. On the other hand, for monocyclic disease like vascular wilt and other soil-borne diseases, it is of 0.02 units per day. And in case of polycyclic diseases like multi-year diseases, as for example Dutch elm disease, cedar apple rust, etc., the rate of inoculum increase for such type of disease is 0.02 to 2.3 unit per year. vertical resistance is governed by, out of these four options, the correct answer is mainly single gene, or that is monogenic, and in few cases it may be few gene for the oligogenic. So related to other options like many genes, that is polygenic, that is a polygene is a member of group of non-epistatic genes that interact additively to influence a phenotypic trait. Thus, they contributing to multiple gene inheritance, that is polygenic inheritance or multigenic inheritance or quantitative inheritance or horizontal resistance or non-specific resistance. These are a type of non-Mendelian inheritance as opposed to single gene inheritance. It is a type of resistance that is influenced by many genes, each having a minor effect. Another option, that is plasmid resistance, it refers to the ability, especially for the bacteria, to resist specific antibiotic or the metal ion. Resistance, or say R plasmid contain a gene or say number of genes that help a bacterial cell defend against environmental factors such as poisons or antibiotics. Plasmid mediated resistance is the transfer of antibiotic resistance gene which are carried on plasmids. Resistance plasmid cannot get inserted into the bacterial chromosomes. Next question is on cross protection is carried with. There are four options. And before answering that question, you should recall the meaning of cross protection. That is a type of induced resistance developing the plant against, say, viruses. And its basis is the prior infection with one virus affords protection against closely related one. So the correct answer is mild strain of same virus. This is a very simple question. Gene for gene hypothesis was proposed by, out of four options, two options are correct, that is the name of scientist are correct, but year is incorrect. So option, if you see options A and D, out of these two options, one option is correct, but we have to look to the year, it is 1942 and 1943, and the correct answer for this question is H.S. Floor, that is Henry Harold Floor, 1942. And some more fact about gene for gene hypothesis is that it was put forwarded by H.S. Floor in 1946 based on his observation on flax, that is linseed rust caused by Melapsora lini. The gene for gene hypothesis states that for each gene controlling resistance in the host, there is corresponding gene controlling pathogenicity in pathogen. The resistance of host is governed by dominant genes and virulent of pathogen by recessive genes. The genotype of host and pathogen determine the disease reactions. This is also a very simple question. The disease occurs irregularly in moderate form and confined to a particular area is called, and the correct answer is endemic. On the other hand, sporadic refers to a disease that occurs frequently and irregularly. And regarding epidemic, it refers to the occurrence of disease in excess of what is normally expected. And the meaning of pandemic means that refers to an epidemic that is spread worldwide or across large geographical region. This is a very interesting question. The virus that infect the fungi are known as, the correct answer is mycovirus. It was Halling in 1948. For the first time he reported mycovirus on cultivated mushroom, that is Agaricus bisporus. And Magnaporde oryzae is the first phytopathogenic fungi in which mycovirus was found. And most mycovirus have double-stranded RNA, but other has single-stranded DNA or single-stranded RNA or double-stranded DNA. So we have to remember that mycovirus infect a large number of diverse fungal species, but considering their prevalence, relatively few high quality genome sequences have been determined. For most mycoviruses, the virions not necessarily kill the fungus but will alter the metabolism of the fungus, resulting in reduced virulence. An almost universal trait of the double-stranded RNA mycovirus is the presence of capsid, which are selectively porous protein compartment in which the genome and replicas are contained. Next question is on dash initiates synthesis of nucleic acid molecule in PCR by specific and complementary pairing. There are 4 options. Before answering the question, let us understand the meaning and use of all the options. First option is DNTP, means deoxyribonucleotide triphosphatase. These are the artificial nucleotides used in the PCR to synthesize new DNA strand, much like DNA replications. DATP, DTTP, DZTP, DTTP are common nucleotides present in the PCR master mix. So, as we know that the PCR, that is called your polymerase chain reaction, which is a technique to amplify DNA segment in a test tube. Here, ribosomes are cellular structure that synthesize protein from messenger RNA or the mRNA molecules. And here ribosomes do not have a direct role in the PCR, but they may be involved in the expression of the genes that encodes the enzymes and the factors needed for PCR, such as DNA polymerase, primers and nucleotide. And the important function of the ribosome include it assembles amino acid to form protein that are essential to carry out cellular functions. And the DNA produces mRNA by the process of DNA transcriptions. The correct answer for this question is primer or tag polymerase. That is option either B or C are correct, or both of them are correct. As we know, PCR is based on using the ability of DNA polymerase or the tag polymerase to synthesize new strand of DNA complementary to the offered template strand. And tag polymerase, it always requires a primer for initiating the replication. This is another question. Quantity of spray in ultra low volume sprayer is, out of the 4 options, the correct answer is 5 to 50 liter. For more details, you please see the table, especially for high volume sprayer, low volume sprayer, ultra low volume sprayer, what is the quantity of spray required, then droplet size as well as synonyms. So the quantity of spray required for high volume sprayer is more than 400 liter per hectare. In case of low volume sprayer, it is 50 to 400 liter per hectare. In case of ultra low volume sprayer, it is 5 to 50 liter per hectare. And this was in the question. And droplet size for high volume sprayer is 100 to 400 micron. In case of low volume sprayer, it is 80 to 120 micron. And on the other hand, in case of ultra low volume sprayer, it is around 35 to 70 micron. And this ultra low volume sprayer, its synonym is dilute spraying. On the other hand, for low volume sprayer, the synonym is concentrate spraying. And in case of ultra low volume sprayer, the synonym is controlled droplet application or CDA. This is the last question of my series, that is on genera which is not associated with rust. The correct answer is C, Syntrachia. And Syntrachia is a species of the fungus that belongs to the family Astrelaziniaceae, and it is responsible to cause smart disease of many agricultural crops. It was first described by Mary Maxime Cornu during 1883. Related to the other options, say for options A, Phacospora, it is an obligate biotropic pathogen that causes Asian soybean rust. It has been reported to affect up to 31 different plant species that belongs to 17 different genera under natural conditions. In case of option 2, that is Ravenelia, they belongs to the family Raveneliaceae that is responsible to cause rust in many agricultural crops, and they belongs to the order Puccineans, and the family contains around 26 genera and about 323 species. The genus Ravenelia represents the third largest genus of the rust fungi and parasitized a great number of leguminous shrubs as well as trees. On the other hand, this Ceratoileum, it is responsible to cause rust of fig, mulberry, and it has number of synonyms like Malupa fici, Phacospora fici, Phalospela fici, and Urodo fici. With this, I conclude my series on memory-based ASRB NET 2023 question paper with explanation. I thank all of you for watching my video from series 1 to series 6. I hope you got lot of knowledge through this series. If so, don't forget to like, share and subscribe my channel. Thank you.